have mostly everybody, and so we'll get started with the afternoon so we can try to stay a bit on time. I'm Lauren McIntyre.
I'm in the way.
Nothing changes.
On Spencer's timeline, I arrived during the program project training grant. And was finished before the launch of the first Summer Institute. So the first Summer Institute happened the summer after I defended. I actually, we were talking about this, all of us defended our PhDs in this room many moons ago. Mine on Groundhog Day didn't resurrect, come back and bite me 6 weeks later, whatever. It is my pleasure to introduce Bruce to those of you in the room, the students who maybe, other than seeing the name and having read some of the papers, you know, will have now the opportunity to interact with Bruce like we used to think about when Phil Hill or Jim Crow would come visit us. And I think I will just say a couple of things about the training environment, because it looks to me like some of the spirit really lives on. And Spencer started out nicely by talking about the need for having this community. And I think that is something that you can see with Bruce everywhere that he has been, is that he really is very inclusive in bringing people in. And the other thing is really very open about what he knows, but what he does not know, and very interested and curious, and curiosity and interest driving interactions with a wide range of people. And when I was a student here with that program project grant, was really a powerhouse with Major Goodman, Ron Sutteroff, Trudy McKay, as kind of these cornerstones, and the and Shabang. Well, Shabang was a postdoc at the time that I was a student. I'm a little bit older.
Never admit that.
But that community, that open community, really has carried forward throughout our careers. I think the people who were part of that program project grant and part of those interactions, really, we've spent a lifetime and a career crisscrossing with each other, and really in a spirit of curiosity and engagement that I think Bruce really fostered. And so if you don't, we could talk about all of the honors and all the awards and all of the other things, but maybe I think for me, the thing to really know about Bruce is to know that he's as interested in what you have to say as I think we are all going to be interested in what he has to say next. And I am super excited to see this next, and how related we are.
Thank you. So it's fun to be back in the family. Or as we would say in New Zealand, back in the whaka.
Back in the who?
Back in the way.
So I'm living in New Zealand. And there's been quite a resurgence of awareness of Maori culture. It's been very pleasing to see. So every time you turn on the news starts with the phrase in Maori. You're expected to begin and end every email with a Maori greeting salutation. My wife's great-grandmother was a Maori. So Beth and I have become aware of our extended family connections. And we met one of the senior members of her iwi, her tribe, who told us that he could recite his whakapapa, the oral history of his family, 23 generations. Think about that. Going back to the arrival of the Maori in New Zealand around 1200. We don't know how accurate those are. They were oral. There's some there's some recorded history in carvings and the facial tattoo but it's a very important concept to Maori. So my friend Philip Wilcox, who was a graduate of NC State in forestry, is seeing how they could exploit this knowledge of family relatedness into current genetic studies. We don't know how successful that will be, but it's interesting to live in a community where family relatedness is key. Don't know how to work this thing. There's not a button on this. Okay. So that's the oral tradition. And the last few years we've seen a growth of genetic genealogy, where current typing techniques give us very detailed measures of relationship. So this is just all these when you get your hey Dennis, good to see. So you get these information about your new relatives based on centimorgans. So centimorgans is a measure of distance along the DNA, along the chromosome, and it gives you some indication of how related you are to somebody else. How much of your DNA do you share measured in centimorgans? So like most of you, we've sent our saliva off to the testing companies. So 23andMe sends a regular email. So recently they said 22 people who share DNA have joined in the last 90 days. And the predicted relationship of these people was third cousin. And one of those apparently was called Valerie. I don't know who she is, but we may share a set of great-great-grandparents, or we could be in different generations. We could be some kind of a cousin. So we share less than 1% of our DNA. It's not very much. This is amazing. So we can get that level of precision. What I'm going to talk about today, we don't get anywhere near that level of relatedness precision on our single marker analysis. So this is clearly the way to go to discuss relationship. I got an email yesterday telling me about my new fourth cousins. I probably won't pursue that. I really can't. There we go. So this is based, it's all empirical. So you start down here with yourself, and you read across here first, second, third, fourth, fifth cousins. And this is empirical information. Just the average amount of centimorgan sharing your first cousin has with themselves. And this is, you can't read that, probably 866 that's not, I forget where the decimal place should be. It's 866 things, but the range is 300, 400 to 1400. So this is empirical. It's gives you some idea of how much even now that there's some imprecision going on. But here, great-great-grandparents don't have many respondents. So all this is based on people whose relationship is known to them, and they send in their SNP profiles. So empirical measures of relationships. I want to talk about putting numbers on relationship. So we have a couple of concepts. So we start, the field started out with correlations, civil rights work on the correlation. You could get correlation of allelic state. And then in 1948, we had this new idea of identity by descent. The French mathematician Malacot said that two alleles are identical by descent if they have a single ancestral allele. Somewhat vague about when that happened. If you trace back the alleles far enough to the beginning of time, of course, all the alleles have a common ancestral allele, but there's some arbitrary cutoff point, and we're going to try and get around that lack of information. So we've got a concept of IBD, identity by descent, and then two individuals have a co-ancestry coefficient, and we have to be careful how this is defined. If we've got these two people, each carrying two copies of a gene, two alleles, the diploids, we're going to take one of those at random from each person. So what's the chance that those two alleles are identical by descent? And this is just simple counting methods. Look at the pedigree. So the way is to find identical twins have a occurrence history of 1/2. And then you get down to unrelated people. So we've got various parent, child, siblings, and so on. So that's a convenient numerical definition of relatedness. If you know the pedigree, you can predict the relationship. These are probabilities. The inbreeding coefficient is the occurrence history of those two parents. So it's like an allele at random from each parent and look at that pair of alleles. And if they were uniting alleles to form an offspring, that's the inbreeding coefficient of that offspring. So that's fine, got a pedigree, we can make a prediction, but these are probabilities. And what does that mean? In this context, it means it's the average over all replicates, all conceptual replicates. So Beth and I have 2 children, same process, different outcomes. And had we continued having children, they'd all be different, right? So although they all have the same pedigree, their actual degree of relatedness can change. And this in the following sense. So the simplest one I can think about is half siblings. Half siblings are people who have one parent in common. So they got 2 alleles each, and one of those 2 alleles comes from the same parent. And half the time, that allele from the common parent is the same allele. So we've got 2 alleles from the half siblings chosen randomly. So it's a 1/4 chance those 2 alleles are from the same parent, and then a half time they're identical by descent. So the occurrence history coefficient is 1/8. Now, that's the prediction, that's the probability. For any particular position on the genome, the 2 alleles from the common parent are either the same or not. So the actual identity by descent is either 0 or 1. The average over all the possibilities is 1/8. So Bill Hill and I worked out a standard deviation for the human genome, and for half siblings, it's quite high. So it's as we get further and further apart, the occurrence histories get smaller and smaller, but the coefficient of variation gets bigger and bigger. There's more and more uncertainty. So that's why the methods I'm going to describe cannot distinguish 3rd, 4th, 5th cousins. So we've got pedigrees. If we have the pedigree, we can make predictions, bearing in mind that those predictions have uncertainty. Most of our studies don't have pedigrees. If we're studying natural populations, even if we're studying maize, and Dahlia was on a nice paper several years ago with Ed Buckler saying, we've got records. We know the history of our inbred lines. They're not always perfect, those recorded histories. So even in our best studies, we might think we know the pedigree, and we probably don't. But most of the time, we don't know the pedigree. So we cannot predict the relatedness, but we can estimate it from our genetic data. So 23andMe, for example, uses 650,000 SNPs, pieces of information, to work out their shared centimorgan basis. And most of our genetic studies have many more SNPs than that. So I was part of the TopMed project, and we celebrated the discovery of the 1 billionth SNP amongst our participants. There's a lot of data. What can we do? How can we translate that into So we're going to start with numbers. First of all, we recognize that any position in the genome that we're going to call a single nucleotide variant or a nucleotide or a SNP, or give it some name. And at that position, the parent transmits one of 4 allele types, A, C, T, or G. Most of the SNPs we study only have 2 variants in the population. So we need to designate one of them as the allele of interest. It might be the most common one, it might be the ancestral one, it doesn't matter. We're going to have some rule for saying how we're going to label these 2 alleles. And I'm going to say one of them is our designated allele, called our capital D, and the other allele is small d. And we're going to quantify the data as a dosage number, 0, 1, or 2. 0, 1, or 2 copies of big D allele. And if you look at tables of genetic profiles, you get this big table of 0s, 1s, and 2s. I'm completely incompetent. Wonder if this one works.
I don't think that's gonna work for, it
Okay.
works for PowerPoint, it might not work for.
It's wrong button. So here's some data. This is from a very nice study, the first successful association study for adult onset macular degeneration. 96 people with the disease, 50 controls. They were typed at a small number of SNPs, and lo and behold, one of those SNPs turned out to be the causal variant, causes of the disease. So the data, we've got the 3 genotypes at the SNP, the people without the disease and those with the disease, and I'll just change that into percentages. So you can see a clear difference in the frequency of the capital D allele amongst those 2 groups of people. And that was sufficient to get a paper in Science. And moreover, we know what that SNP does. So it's worked. And that gave rise to a lot of hope and a lot of success, of course, since then. So that was a study, a random study of people with and without the disease. Most of our studies have a lot more than 140 people. We're now seeing publications based on 1 million people or more. So those individuals are not independent. Who would have guessed, but they're related to some extent, either because of recent common parents or more distant evolutionary timescale relatedness. So those dependencies amongst the people are going to have a big effect on our association mapping studies. And almost anything we do is unlikely to succeed if we ignore their relatedness. So just one of the many reasons why we're interested in this topic. So the first approach has been to look at not identity by descent, which almost always we cannot observe. We don't, by looking at the alleles, we can see what type they are. We don't see their history, but we can see the type, identity, and state. So we might characterize the relatedness of these 2 people. So they've each got 3 genotypes, 9 combinations. We just count the number of pairs of alleles they share, 2 pairs, 1 pair, or 0 pairs. And very often we can describe, this is going back a long way, Elizabeth Thompson had a classic paper in 1975, where she worked with 3 coefficients, probabilities of 2 individuals carrying 0, 1, or 2 pairs of alleles identical by descent. That little table of co-ancestry coefficients I showed you for siblings, parent, offspring, and so forth, that co-ancestry is it's a summary of the 3 kappas. It's just an average of those 2. So it'd be nice, for example, to get started if we could estimate those kappas. And here is a way of doing that. This is sort of the standard way. It's in many packages, including the Plink analysis package. So we've got a large number of SNPs. Let's say we've got 1 million, and we count how many of those of the SNPs do 2 people share 0 or 1 or 2 pairs of alleles that look to be the same, and we just simply take the fraction of SNPs for which they share no alleles. So they're the opposite homozygous. This person's big D, this person's small d. So that contributes to the L0 number, and the proportion of SNPs in that class leads us towards an estimate of kappa 0. There's like a normalizing constant here, and this is the sum of the products of the allele proportion. So I like to use tilde for a sample value and keep P for probability. So P tilde L is the proportion of the designated allele for the Lth SNP. It also uses this other one, which is the product of the squares. So we can get simple moment estimators, 3 categories, 3 counts, everything can be solved, and we can summarize those estimates by giving us an estimate, a moment estimator of the current ancestry coefficient for unrelated, non-inbred relatives. So that's old, that works. And here are some results. We had a study, and the manifest for the study gave us all the people's genetic profiles and indications of their relatedness. So we used the Plink package, estimated kappa 0, kappa 1, kappa 2 comes by subtracting from 1. And here's the cloud of people who told us they were full siblings. And indeed, those estimates are clustered around their expected values of kappa 0 of a quarter and kappa 1 of a half. So there's the cloud of full siblings. Not all are exactly at their expected values, of course. We've got the variation imposed by genetic sample. Here are the people that look awful like parent offspring. Kappa 0 is 0. Parent and child must share one pair of alleles. But here are some people where kappa 1 is not quite 1, it's less than 1. So kappa 2 is bigger than 0 because these people, the offspring had, the other parent was related to the parent of interest. So these are for pairs of people, and the missing parent was related to the first parent. I said that very badly. The people and the offspring in this collection of people they had related parents. And here are the identical twins. Here are the half siblings and various degrees of cousins. So there's the estimates, and these are widely used there, easily obtainable. So statisticians got involved, and they said, well, this is we can do that. We can handle count data. We've got binomial variables. We've got 0, 1, or 2 copies of the designated allele. Let's call that the dosage for SNP L and person J. And moreover, we know about these. We've got a sample of people, and if we add up all their dosages, we're getting twice the number of big D alleles in that set of people, because the X's are counting the number of D alleles. So we add up all the X's, divide by the number of people, and we get their average, sample average, right, which is indeed the sample average of twice the sample average of the designated allele. That's SNP. So we know the mean, and we write down those dosages in a matrix. We've got rows for people and columns for SNPs. We've got the observation, we've got a sample mean. Why don't we do the standard thing and divide by the sample standard deviation? And it's binomial, so we know all about variances, standard deviations. So let's build that ratio. And we might divide by the number of SNPs just to keep things manageable. So I think one of the first times this appeared was in the Eigenstratt papers by Patterson and others. It's a classic dairy science paper by Van Reyden, and then more recent work in the GCTA package. This is a standard statistical way. No genetics here whatever. No thinking about genetics at all. We've got count data, and we just manipulate it to standardize it. And the intention is that this matrix of elements for the 2 different people should give us estimates of the inbreeding on the diagonal, or technically 1 plus the inbreeding, and co-ancestry, or twice the co-ancestry off the diagonal. But they don't. So most of the packages that use that package are not telling you what they purport to. So I want to just go through the method we've been working on, and just the spoiler alert, we're going to get to the end saying, I'm showing you newish work, but we were beaten to it by several years, and it's a funny story in retrospect, but not funny at the time. So let's talk about the probability a person's a homozygote. And that person has an inbreeding coefficient of capital F. That's the probability that person's alleles are identical by descent. So if they're identical by descent, they're the same type, So the person's homozygous. Otherwise, not identical by descent. The alleles are independent, and we've got 2 copies of the designated alleles and 2 copies of the other allele. And I'm going to use pi as the probability an allele is the designated type. So that's a prediction of the probability. So there's the probability and an inbred individual is homozygous. So that's fine. 2 problems with that equation, of course. I want to estimate if I don't know it, and I don't know pi, and we would say pi is not knowable. It's a probability, it's the average over an infinite number of replicates of an evolutionary process. We could think about it as the allele proportion in the reference population to which identity by descent is defined, but let's not even worry about that. Let's just say we don't know it and we're never going to know it. It's a nuisance parameter. Let's do the same thing for 2 people, and it's an abuse of language, but let's just call that homozygosity in quotes. What's the chance that 2 alleles, one from this person, one from that person, are the same type? So that depends on whether those alleles are IBD, it's a co-ancestry, and we've got the same equation. So now we've got 2 equations linking the parameters of interest, the f and the theta, the parameter not of interest, and we don't know it, but the left-hand sides are things we can observe. We can observe the allelic state of these 2 alleles. So we'll get a sample value of, we could count the number of SNPs that a person is homozygous or that 2 people share an allele. So can we manipulate those observations to estimate f and theta? So we could, we've gone through various phrases. We're now calling it allele sharing. So alleles are shared when they're the same type. So we've got our 3x3 matrix again, the 3 genotypes, and we're going to put in the proportion of alleles those 2 individuals share. They're the same type, identical state. And there's the 1, the 2 homozygous, and the 0. All the other terms involving heterozygotes are a half, because we're talking about 2 alleles taken randomly. So this is a little bit different from that identity by state matrix. It looks like a small difference. Anyway, it is what it is. So we've got these allele sharing statistics, 0, a half, or 1, for each pair of people at each SNP. And we'll call that the allele sharing number for 2 people. And then we're going to want the average allele sharing for all pairs of different people in our study of N people. So we've got an average allele sharing. This is the allele sharing for a subpopulation. Why do I use S? Because Sewell Wright used S in 1922 for a subgroup. But now we've got allele sharing for a person. The homozygotes share 100% of their alleles, so their allele sharing statistic is 1 or 0. This is just a fancy way of saying person's homozygous or not. And then for our study, we'll take the average allele sharing for each person and call that the individual's I. So we've got those simple observations, and we're going to want to combine information over SNPs. So we'll take the average allele sharing for 2 people or for 1 person. So those are our building blocks, our statistics. Now, what are those things? How do they help us? Well, we have simple expectations. Expectation meaning in the genetic sense over all replicates of the history of those pairs of alleles. The allele sharing for 1 person depends on that person's inbreeding coefficient, and averaging depends on the average inbreeding coefficient for the study. The allele sharing for 2 people depends on their co-ancestry, the average over all pairs of people on this population-wide allele sharing. So those are the parameters of interest. The observed sharing with the tildes, those are observations. Here are the parameters. We've got a nuisance parameter, capital A, which is the average of the sum of the products of the allele probabilities. We cannot know that. So we need to get rid of that quantity. In retrospect, this doesn't look all that pretty. If we had have done 1 minus A, we wouldn't had to have 1 minus on the left-hand side. So I could have called it allele non-sharing, I suppose, or heterozygosity. It's a thought. Multiple SNPs. 1 SNP's not going to help us. We need a lot, and and I say we. This is Clark a long time ago. Clark Hoggerham said, let's when we form ratios. When we I must have left out a slide, did I? No. Never mind. I'm going to, here's my estimator for the inbreeding coefficient for a population. I'm going to take the allele sharing for average over individuals, average over pairs of individuals, and I'm going to add over the SNPs. So I've taken the ratio of averages. I've got the sum, but I could have divided by the number of SNPs and got the average. So I've got the ratio of averages over SNPs. Now, this, we've had several rounds of papers that have been reviewed by our peers, going back to my work with Clark and my recent work with Jerome. And almost every time the reviewers say, particularly if they're statisticians, they say, don't do that. You can't pretend the ratio of expectations is the expectation of the ratio. That's classic. That's stat 101. Don't do that. At least give us some, at least acknowledge that. Say something about it. Just don't ignore the issue. We used to say things to the extent that. Anyway, and then we were rescued a couple of years ago by John Story, Ochoa, who went through the very simple mathematics to show the central limit theorem does indeed apply to statistics of this form, and there are very mild conditions on these statistics which are satisfied with that for our work. This ratio is asymptotically unbiased for exactly what we want. Asymptotic in the sense we need a lot of SNPs. It has nothing to do with the sample size. The number of individuals is almost irrelevant. We are going after genetic sampling. And the genetic sampling is addressed by having replicas of the process from our different SNPs. The statistical sampling by the people we sampled has very little effect on anything we do. So here's our estimator of the inbreeding coefficient, what we wanted, the average for the sample, the population, and this is IBD for a person or an average over a period. And we need to, that's an absolute, that's a probability. We cannot interpret it unless we put it in context. So we say this person is inbred because the chance that they have IBD alleles is big compared to the chance that 2 alleles in different people are IBD. So we put things in context, and we have to standardize things. So we end up with what Civil Wright called FIS. This big one is FIT, and this one is FST. So we've got the within population inbreeding coefficient. So I think Dalia's been using my example, but I'll say it again anyway. But just
They won't have heard it.
so I like to talk about a large family reunion somewhere in the Midwest where the families are large, and there's a family, I'll call them Smiths. So a lot of people come to this family reunion. They're having a great time, and they say, "No, this is great. We're related. So we've got good qualities. We're good people." And there's a lot of first cousins in this group who are unmarried. So they decide to form their own community. They do that. They go off and pair randomly and have children. And then Henry comes along with his little test tubes and asks them to spit into the tube for genetic testing. And he didn't think about it. He said, well, you know, these people, the offspring, their parents are all cousins, so I'm going to find some inbred. He sends it away to 23andMe, and it comes back. He does a Hardy-Weinberg test and get perfect fit. No evidence of inbred. So what's the disconnect? Henry's standing in that community, and with respect to that community, the people are not related. They're all equivalent. There's no pair of parents different from any other pair. So essentially unrelated. And I did slip in that you didn't challenge me. I said the parents mate randomly. Of course they didn't, but essentially, their children are not inbred because their parents are unrelated. So within the population, there's no inbreeding or relatedness. I'm standing outside Henry. Those people are all cousins. They have a current history of 1/16. All their children are inbred. So we're both correct. It's a matter of perspective. So if you've only got data from the one population, you cannot detect relatedness. You've got no comparison. We can compare each pair of people to every other pair in that community, but in our example, they're all equivalent. So there's no current history, no inbreeding within the population, but from the wider perspective, of course, there's a lot. So, and that was the essence of a civil rights introduction of correlations of pairs of alleles in a subpopulation relative to the total FST, or pairs of alleles in an individual relative to the subpopulation FIS, or pairs of alleles in the same population relative to the total FST. So there's our allele sharing estimate. It's asymptotically unbiased. It's very simple. And look, sample allele frequency does not appear in the estimate, and in the expectation, pi does not appear. So we've gotten rid of that need to know the reference population or the allele probabilities. So we have an estimate. We depart here from civil right and do it for each person. Each person has an inbreeding coefficient of interest to us. We cannot estimate that quantity, but we can estimate it relative to the average current history in the population. So that's the parameter, and here's our allele sharing estimate. So notice the duality between the IBD parameters on the top line, and here we've got the allele sharing statistics. So for every pair of alleles, there's a probability that they're identical by descent, and for every pair of alleles, there's an allele sharing statistic based on the data. So that's classical, that's 1922, but we did a very trivial extension, and we haven't found the word for it, but let's do the same thing for two individuals. Their co-ancestry, which we'd like to know, let's measure that relative to the average co-ancestry and standardize it, and we'll call this the within population co-ancestry coefficient for those two individuals. And so that's the parameter, the compound parameter of interest, and here's the allele sharing statistic. By construction, these statistics, by construction, these sum to zero. So we're going to measure co-ancestry. Some pairs of individuals are more, some are less related than on average for that population. Individuals are inbred if their allele sharing is more than their alleles are more likely to be the same than they are between people. So sometimes this ratio is positive, sometimes it's negative. And that causes some people to worry, but we'll come back to it. So having got this far, why did we do it? Why wasn't the old way good? And what about those standard methods and the kappa coefficients and so forth? Well, our sample allele frequency is indeed unbiased for the population probability, the allele probability. But when we square it, of course, the expectation of a square is not the square of the expectation. And we've got terms, we've got the pi's, we've got what we thought we might be estimating, but we've got the average kinship, average co-ancestry for all the pairs of individuals in the sample that gave us the sample frequency. We've got the average inbreeding, and here's the sample size. Let's forget about that. Let's suppose we've got 100 people at least, or 10. We have a large enough sample, we can ignore this term. So the expected value of p squared is what I squared plus this other term. And the expected value of this product is not 2 pi 1 minus pi, but 1 minus theta s. That's not new. Even I knew that back in 1996. Going back a few years before that, here's the, I think one of the first estimators for inbreeding coefficient by Leon Horvitz, 1953. And this is for the sample as a whole, the population. They said the inbreeding coefficient for the individuals in that population is 1 minus their, they use heterozygosity, and now notation is just the 1 minus the allele sharing divided by twice p tilde 1 minus, put in this product of the sample allele frequencies. So if they had have known, if we knew, if they had have known the allele probabilities, they had to put pi's in here, this would be a wonderful estimator. We don't know pi, we put in the sample values, and as a consequence, what we're estimating is not F, it's this thing, it's the within population inbreeding coefficient. It's Wright's FIS. So it's fine. It's not wholly bad. We really want to know about inbreeding. Now, for each individual, we've measured it with respect to this theta rest, this average current history. But this term here and here, those are applied to every individual. So whatever ranking there is for the inbreeding coefficients, the same ranking will apply to this compound parameter. So we are ranking individuals or populations by their inbreeding coefficients, even without knowing the inbreeding value itself. Come back to that. So here's the standard inbreeding coefficient. There are various forms of this, but it's and I've written it as the ratio of averages. And the original publications had the average of ratios, and there's the inbreeding and the co-ancestry. What is this thing estimating? When you use Plink or GCTA or almost any package, and you say estimate the inbreeding coefficient of these people, they're not estimating capital F, because that's not possible. They're not estimating the within population inbreeding coefficient either, because they had these P's on the bottom line, giving that bottom line, which looks familiar, we're happy with that, but they've got a P squared on the top line. So the expectation of the square involves the variance of the sample allele frequency. So what they're estimating is inbreeding relative to this thing, and this thing is the average co-ancestry for individual J. I've got this set of individuals, each with an inbreeding coefficient, each with a co-ancestry for every other person in the study. So I've got an inbreeding and I've got an average co-ancestry. So I'm estimating the inbreeding within population inbreeding relative to modified by the average co-ancestry. So whatever ranking there is for the Fs does not apply to these estimates, because individuals do not all have the same value of the average co-ancestry. So we've lost the ranking. And similarly for the co-ancestry estimate, the standard estimate is what it certainly cannot be what we wanted. It's not even what we would accept. It's got this modification. So we're talking about identity and state. And identity by descent. Identity by descent means these two alleles have an ancestral allele. We don't observe that, but we get their type, we get their identity and state. The co-ancestry and inbreeding coefficients are probabilities of identity by descent. Cannot estimate those, but we can work with the data and come up with quantities which at least have the same ranking as the things of interest. So let's look at the data. So those of you who haven't seen the 1,000 Genomes Project, there's a little example, little illustration. It's a very nice public data repository. 2,500 people from around the world typed that a lot of SNPs. We've been working with 77 million, but there's many more available now. We've got some African samples and Afro-Caribbean and African-American. We've got some Native American, got some European, some South Asian and East Asian people, all self-identified. all completely typed, very nice data, no missing SNP data anywhere in the collection. And here are their names. They've all got, they are lumped into groups. The 1,000 Genome Project, I think at one time just called it meta population. We're just going to call it groups of roughly corresponding to continental ancestry. And then just to put things in context, we know we've got a good model anyway for our own history, an origin in Africa, and a migration north and east over time. So the further away we get from Africa, the longer it has been. So the more differences we will accumulate between populations at that level. So this may be the most important picture we've developed. For each of those 2,500 people, we've got the allele sharing estimate of their individual inbreeding coefficients relative to the average co-ancestry of all the people in the study. So we've got this, they've got the same reference set. We've got 2 alleles per person compared to all the pairs of alleles anywhere in the study from different people. So here we've got the African people. We've got these ones, I think, the continental African. They've got the African American and the Afro-Caribbeans. Here we've got the Native Americans, the Europeans, the East Asians, and the South Asians. So inbreeding average co-ancestry, and we see a general positive trend. What we do not see on this plot is any indication that there's no inbreeding. Not all of X equal to 0, or no co-ancestry. So any paper that has an estimate of inbreeding coefficient that assumes the people in the study are unrelated, as did Lee and Horvitz, is not going to work. Any study of relatedness that says that people are not inbred is not going to work. So keep that in mind. This is data. There is inbreeding. All our natural populations, humans in particular, are inbred, and they have related people on them to varying degrees. Now notice we've got some negative numbers here. And if you don't like negative estimates of inbreeding, that's fine. We can just slide things along. Clark and I did something a little sneaky. We said what we were doing. We got rid of those troublesome negative numbers by saying, let's imagine that the populations we were sampling were independent. That's one way around the problem, and then I'll come back to that. Ochoa and Story said, well, we're estimating inbreeding or co-ancestry identity probabilities. We don't like negative numbers at all. We're going to avoid that problem by saying, in our study, the least related people have 0 relatedness. So let's shift the goalpost, make our smallest value equal to 0. I don't think that solves the problem. The problem has no solutions. And here, just to look at the South Asians, the East Asians, and the Europeans. So we've got different ancestries have different relationships between inbreeding and relatedness. It's the South Asians with communities which somewhat encouraging of cousin marriages. So we get more inbreeding than relatedness. Now, here are the individual inbreeding coefficients for the 2,500 people colored by their continental ancestry. Here's the African, the Native American, I think this is the East Asian, European, and South Asian. And each dot's a person, their inbreeding coefficient relative to their own population. We say the allele sharing each person has compared to the allele sharing between pairs of people in their own population. So not much difference on the average over the world. You know, we're all people, we all obey the same general societal rules about marriage, about marriage rules. So there's not much inbreeding. There are some high values. There are some, you can't see the scale. I can squint in the, on this scale is 0.5. There are some people who are highly inbred by this measure. There are people, and it's more pronounced in the UK biobank data, which is a much bigger sample, 500,000 people. There are people walking around quite happily who are clearly the offspring of first-degree relatives. Now, putting things on another perspective, let's take each person now and look at their allele sharing compared to any two people anywhere in the world, the ones that I was plotting on the previous plot. Now the Africans look very different. They are clearly much less inbred than anybody else in the world. There's so much more diversity in Africa and African populations than anywhere else in the world. The people are less related than other people. Everybody else is, the non-African people in the world they represent a small fraction of African diversity at the time of the migrations. And so these Native Americans are highly admixed populations, as complicated mixtures of African, European, and Native American. And we've got the two Asian and the European. So these all look different now. But here is the South Asians. So those are the inbreeding coefficients. It depends on your perspective, and this is maybe the most informative one. Now that was inbreeding. Here it is for the co-ancestries. So here we go, the allele sharing estimates for each pair of people relative to pairs of people in their own continental group, their own group. Not much difference in relatedness across the world. But when we compare two people to any two people in the world, the Africans are clearly less related than anybody else. Here are the standard estimates. These are the ones that you will invariably get from using any of the current packages. You will get evidence that Africans are more related than anybody else. It's not true biologically. And this is a consequence of these not being estimates of co-ancestry. Co-ancestry modified by the average co-ancestry of each of the two people. So we've lost the rank. So let's go back to 23andMe, and they're using they are in effect observing identity by descent. They are single step methods, they're not. So the idea is very simple. You look at the genome, it looks like a single person. Look at the genome of that person. As you go along the genome, if you get a run where every SNP's homozygous, and we're talking about a run of centimorgan or more, 1 million bases or more, a lot of adjacent SNPs are homozygous. The only plausible explanation for that is that those two chromosomal chunks are the same. They have the same history. They're identical by descent. So it's a direct observation of regions of identity by descent in a person or in two people. We look for regions of the genome where there is so much similarity in those two regions. In the two people, the only plausible explanation is identity by descent. So you look at the fraction of the genome that's in these regions of identity by descent inferred, and say that's the proportion of the genome identical by descent. And that's why the methods give us these very good estimates of distant relatedness. It's not error-free. You have to say, well, how long is the region? And how much wobble do you allow? How many heterozygotes do you allow for this? There's some tuning to go through. All right. If you adjust the length of the region that you require to be met for homozygosity or sharing, in essence, you're adjusting the time backwards that you're looking. So very long regions can be the same because of recent ancestry. So you're looking at very close familial relatedness. You don't look back, you look at long regions. If you allow shorter regions, you're looking back at the consequences of a much longer history. So it's a different perspective. We've really changed the question. It's identity by descent. with respect to this definition of the length that you're looking at. The methods I've been showing you, the single snip methods, are looking at single snips. And we're talking about their identity, regardless of whether you're looking back a generation or 1,000 generations. Does it make a difference? Not much. Here are the allele sharing estimates for the 1,000 genomes individuals. Here are the runs of homozygosity estimates we got from Flink. There's this very good concordance, particularly in the Asian and European populations, and then it begins to sort of depart from collinearity. We get some differences, but by and large, we're not going to be misled by using either one. You use the standard methods, you're fooling yourself, because those estimates have nothing to do with the quantity being estimated by the runs of homozygotes. Right, so that was too easy. Clark had a paper in 1971, following earlier papers by other people saying, well, we've got two people, that's 4 alleles. There's all kinds of identity patterns amongst these 4, and we might summarize those by having these 9 patterns for 2 people. X and Y are the people. There are 2 alleles in the top or bottom line, and a line joining identical by descent pairs. So here's 2 people who actually only have 1 allele between them, 1 independent allele. They all have copies. Can that happen in practice with humans? I've got 2 nieces who have children with brothers, their partners were brothers. So these children are double first cousins. They only have 1 allele, obviously 1/64. That happens. And there's the other extreme, no identity. So we've got 9 recognizable classes, 9 pairs of genotypes, including order, homozygous D for both individuals or the small d, 2 heterozygotes, homozygous heterozygote, and then the other pair down here should be. Am I missing 1 pair? Here it is here. So we've got nine classes of genotypes, and we could regard this G as the probability of that class, or for the data, we say the proportion of SNPs that for which the 2 people fall into one of those 9 classes. We can express those in terms of the 9 identity by descent probabilities, and all we need to know is the allele probabilities. So there are 9. It's a 9 by 9 matrix. There are 9 categories. Of course, their 9 Gs must add up to 1. Those are the only possibilities. And there's another relationship that's exists among the rows. So there are actually like 7 degrees of freedom, I guess you might say. I like Clark's notation. I'm biased, but he, instead of working with Jacquard's 9 coefficients, he translated them as summaries of the 2 inbreeding coefficients. X and Y have an inbreeding coefficient. They have got a co-ancestry, and then they've got some new ones. They've got three alleles carried by 2 people, or 2 pairs of alleles, or 4 alleles. So we've got just parallel notation. So these are quantities of interest, and there are publications giving estimates of those quantities. Can they be any good? I mean, would you expect them to work, given what I've said already? Of course not, because we don't know the pies. If we knew the pies, you can invert the matrix. That'd be easy. We don't know the pies, and in particular, we might say, I got a good estimate of pi itself, and not of the square or the cube or the order. So we cannot estimate the deltas. Why do we care? Well, you know, as Spencer said this morning, we started out, and we're still there, thinking about quantitative genetics. So it's very interesting to know the covariance of trait values for 2 individuals, particularly if they're related. So the covariance of their genetic contributions depends on the co-ancestry of the 2 people and the additive genetic variance. So that's classic. I've just written it out. If the trait's additive, then this is a very nice equation. There's another term which brings in the non-additive, the dominance variance. We don't know quite as much about this. But then Clark points out there are three more components of variance, generally ignored, but they're important to an unknown extent. We don't have estimates of these other things. We've got predictions. We know very little about these other coefficients. It'd be really nice if we could estimate the data. And now you would think we have enough data to do that. So the covariance for additive traits, and most traits are largely additive, just twice the current history, and the variance of a trait for an individual is the inbreeding. Now, what's the variance of a trait for one individual? It's the expected variance over trait values if that person's history was replicated under the same environmental conditions. It's a funny thing to have a variance for an individual. Anyway. So if we had a matrix whose elements are 1 plus f on the diagonal and 2 theta off the diagonal, would have what's called the genetic relatedness matrix. And if we've got a pedigree, we can predict these values, we know them, and plant and animal breeding use those predicted GRMs for many years and successfully. But now we say, well, we don't know the pedigree, and we use estimates. Just a little aside. So the thing about estimation, if we sort of has the same approach we've been doing, we'll categorize our individuals as in terms of the allele sharing. All the alleles are shared for that pair of genotypes and for that pair, or they have there's 5. It reduces down to 5 patterns of allele sharing, which is an interesting observation because this guy had a very provocative paper, 2014, says for SNP data, identity by descent measures are non-identifiable. So we can't estimate. And why 5? You might ask, and this might be one reason. So let's go back to the Kappa estimates of Elizabeth Thompson and others, good estimates. If there's for non-inbred relatives, no inbreeding, so instead of so 6 of the 9 deltas are 0. We're just left with these three of them, 7, 8, and 9, which we call Kappa 2, Kappa 1, Kappa 0. And we don't know the pi's, but let's put in the sample values. So now the expected value of those counts, the ones that Plink uses, the number of SNPs that share no alleles or one pair. So what's the expected value of those counts? It depends on all the deltas and H and K. If the individuals are not inbred, every delta 1 through 6 is 0. So N0 has an expected value that depends on Kappa 0 and capital K. We want to know this one. We're a bit nervous about this thing because I don't think we know it. But let's just stick in the sample values, replace the pi by the sample frequency. So there's the estimate, the estimates, and there's the current history. So what are these things actually estimating? Not the kappas for sure. They're estimating these things. Well, but these I said the individuals are not inbred. You can't say that. There's no population there's no population of relatives where there's no inbreeding. Think about it. Not possible. So let's not do that. So the problem is we don't know H and K. And that means that the estimates we use are complicated. If we reduce them to the current history, we're not estimating the current history. We're not even estimating the thing I was talking about. It's a funny quantity. The other problem is there is inbreeding. Suros did a couple of nice things. He said, well, there are 5 identifiable quantities. I can estimate inbreeding at each individual and their currency. So I've got 2 more things. He gave 2 estimates, and I can talk about one of them. It's the proportion of individuals that share 3 capital Ds compared to the proportion that share 3 small ds. So let's do that contrast. You can estimate that. And well, can you? That thing the observed value of that difference depends on the inbreeding, the current history, and only on the 3 allele measures, not on the two pairs or 4 alleles. So it's a complicated thing, but at least it's sort of a measure of IBD that involves 3 alleles. So in that case, it's new, and it involves pi 1 minus pi 1 minus 2 pi. And he called the square bracket thing, called that phi. And then he said, well, I don't know pi's. Everybody just sticks in the sample values. So let me do that. And what is he estimating? Well, he's estimating something that depends on the average IBD for all pairs of alleles in the sample and all trios. So you're not estimating what he was wanting to estimate. And that's it. I'm running out of time. Let me just say, everything I've said was for individuals, 1 at a time or 2 at a time. Let's change the word individual to population. Let's do allele sharing within and between populations to give us FST. We can do that. I don't have to do any it's easy to do. And didn't you do that already? 1984, you had a paper. It's been quite well cited. I'm going to try and destroy that paper right now, right in front of you. You were here. We said that all the populations were equivalent. They had the same average co-ancestry. Clearly not true. And all the population pairs were independent. So the theta between the populations was 0. And under that model, it made sense to take weighted averages over samples from populations. To weight the sample frequencies for population I, and that was the appropriate thing to do under that model. That gave you the best estimates. We worked with 3 mean squares. We did it, I think Clark was an expert in this area, and we in essence we did an analysis of variance of allele frequencies. 3 mean squares, and we can write those mean squares in terms of allele sharing, but using weighted allele sharing. And we had somewhat cumbersome FST estimates, and we also got FIS and FIT as well. What were we actually estimating? What we said we were estimating, this thing, under our model, and we said that. But that's not the reality. If you say, well, the populations are not all equivalent and there's current history between them, what we're actually estimating is close to this if the sample sizes are the same. So if the Weir and Cochrane model was correct, or the sample sizes were the same, we are getting the same expressions as the allele sharing estimates. If they were independent, we would get back to what we said we were estimating in that paper. And to be fair to Clark, he knew that. We didn't make it quite explicit in the paper, but 10 years before, he said, we've got some set of alleles, an individual or a population. We've got some target set. We're comparing it. We're actually comparing it to the average between population density. These are the things that are estimable. And we've got allele sharing estimates. If theta b is 0, which we said it was in 1984, then these ratios are the same as the thetas. Thus, in practice, the model is reduced to accommodate the parameters estimable from the information available. The estimable correlations are always relative to the correlation of the genes farthest apart, generally the least related and the least correlated. While in practice, one drops the distinction between psi and theta, introduction of this model points out the result of enforcing this constraint. So he gave fair warning. But we didn't follow up. And we did allelic data, and it has additional problems. We said, but we didn't stress. If you're going to use our method for data on alleles rather than genotypes, you have assumed hardly one big equilibrium. We didn't say that out loud. We should have done. Does it matter? Not too much. So we submitted our paper, and I've just told some people I've given my papers to the library yesterday to go alongside the Cochrane collection, and dug up all the reviews. Reviewer one, for the 84 paper, said reviewer one felt it was very caustic in places. I'm afraid in its present form, it will invite a lot of correspondence that will cloud the picture. Similar point was made by Peter Smouse. Joe Felsenstein, the associate editor, said evolution should not serve as a forum for an endless and confusing set of replies. And this is what the predictable quarters did. The predictable quarters, Masatoshi Nei, recently departed, worked with heterozygosity, H0, HS, and HT. 30 years later, I can acknowledge that H's are allele sharing statistics. This is not the AS. The AS was alleles from different individuals. AW is for 2 random alleles from a population, including the case where those 2 alleles were the same allele or from the same individual. So the sample size will affect the answer. And he used HT, any 2 alleles in the study, whereas we use HB, any alleles from different populations. His HT used any 2 alleles, including 2 alleles from the same person, or from 2 people in the same population. So it's affected by the number of populations. Modified that in 83. Chester came to visit. It was an uncomfortable set of discussions we had with Ron Chester. But he modified HS to give our one, our sample, and he modified HT, somewhat complicated, and that helped. And then I've got to come right out and say it. In 1987, in Masatoshi's book, he got back to exactly what we do. His estimates are identical to our allele sharing estimates. So were we allowed to be caustic? For predictable quarters, we should not have had that correspondence. It's a great pity that that we'll publish this, and I think we'll have a commentary as well, saying that, of course, there is differences, and we're putting it on a comp. And Masatoshi was not concerned with relatedness or inbreeding; he was population structure, and we've got the individual values. So we knew what we were doing. We said it. We didn't stress it, and the 22,000 people who have cited that paper didn't read what we said in the paper because they wouldn't be doing it. And I hope they will stop citing it, not reading the paper. And Masatoshi's approach was different. He was not concerned about the evolutionary history. He wanted to describe the current populations with simple, easily interpretable statistics, and he certainly did that, which we didn't. But we are actually doing the same thing. We're not estimating IBD. They never said we were. We implied that we were, and we weren't. So this is from our polyploiders. Don't ask me about this, but we've added an employee level, following up on some of my work from my academic grandson, student of Ken Dodds, with similar values. And we've looked at forensic applications. Had a couple of recent papers, one on inbreeding depression this year. So that's all I know about relatedness. How related are we? We're all related. We're all inbred. We can predict those values if we have our whakapapa. We can estimate them relative to a reference set from data. And it's a lot of work remains to be done. So thanks for listening.